It took William Cumming Rose 12 kilograms of protein to get this much pure threonine, but only a few of his grad students to prove it's essential. So threonine is the 6th amino acid that we're going to talk about in 20 days of amino acids. Amino acids are protein letters. There are 20 common genetically encoded proteinogenic ones. So basically there are 20 ones that your cells can tell the ribosomes of the protein making machinery to put these in this order to make a protein. And these amino acids are able to get linked together because they have this generic backbone part, which has this amino group and this carboxyl group, which can link up, and they link up into chains and get folded up to form a protein. And how they fold up is going to largely be influenced by the side chains, so which are the unique parts of each of the amino acids. So these side chains have different properties, and they're going to influence how the protein folds and how the protein functions. Today we're going to look at threonine. Threonine's side chain, you can remember it has like 3 non-hydrogen groups. One of them is an O, so 3-O-nine, that's, you can remember for the O, and then the nine is like 9, and there are actually 9 atoms in the side chain. So that's not where the name comes from. The name actually comes because it looks kind of like this sugar, like 3-O sugar. But it's a helpful way that you can remember what this looks like. And it's important to remember that it has that O group, because that O, that OH, that is going to allow it to act as a nucleophile, so it's going to attack an electrophile like the gamma phosphate in ATP. And don't worry, we're going to go into all of this, but basically it's going to give you a way to post-translationally modify this. So we're going to modify it after it's already been added to a protein chain. So the process of translation is where these amino acids get linked together based on the instructions that are coming from the gene and then the messenger RNA copy of the gene. And when they're linked together, they're linked together as like the state that they were like made in, like their generic state type of thing. But then you can also get what are called post-translational modifications. So these happen after they are linked to the chain. And so one type of the most common type of post-translational modification we talk about is typically phosphorylation, which is the addition of this phosphoryl group. So this it's technically a phosphoryl group and not a phosphate group, because one of the oxygen is going to come from the protein already, so you're just transferring like a phosphorus with 3 oxygens instead of 4, but the end result is you get basically a phosphate onto your protein. This phosphate group is negatively charged and bulky, and so it's going to influence how the protein, it can influence how the protein is like shaped and how it acts. So you can think about it by like creating new binding sites or removing binding sites that may affect the what the protein hangs out with, and it can also influence the protein's fold. So basically it's kind of like if you packed your suitcase and then you realize like, oh, I want to put in this one more item, like maybe you went to a shop in the airport or something, and then you've got to repack your suitcase to try to accommodate the new item. It's kind of like that when you get a post-translational modification. You can have these changes as the rest of the protein tries to kind of like shift to accommodate the new thing. And so these can be like allosteric changes. If you have like the phosphorylation can occur in one part of the protein, and the effect can kind of like ripple through, and so you can even affect parts of the protein that are far away. So you might even have like a protein like opening or closing or doing wild things, and that might affect the protein's activity. So basically when the protein's folding, normally it's forming when it's like the amino acids are in the form that they're added. And so when they're in the form that they're added, then the protein is folding up to accommodate the various likes and dislikes of all of the different side chains. So some of them, maybe they'll be like, the hydrophobic ones will be like, water does not want to hang out with me. I want to hang out with hydrophobic things instead, because the water is going to cinch around me and force me into the center of the protein. And the hydrophilic things are going to be like, hey, I want to hang out with the water, so put me on the surface. Then there are things like the bulkiness. So maybe things are like, dude, I can't bend like that. Or then there are things where the charges, so maybe there's like a positively charged thing. It's like, I don't want to be next to that positively charged thing because we repel each other. He's repulsive to me. And then it may be like, oh, I love that negatively charged thing because I'm positively charged, so I want to hang out with that. And then you can get like a salt bridge or an ionic bond between them. So there are different ways in which these different side chains can interact with one another and with like the protein backbone as well. And this is going to influence how the protein folds. But then if you add a post-translational modification on it, now you're changing it's like you're changing one of the letters, and now all that work you did I mean, it still really matters, but now you can have that work kind of like changed. So this is why post-translational modifications can have these big effects on the structure, as well as effects on creating like binding sites for other things, because now you have like this different group, and so maybe this like another group's this, and it's like, oh, a phosphate group, let's bind, you know? So this OH group, even if you don't like modify it at all, it's going to provide this polarity to this molecule. So basically it's going to provide a partial charge, and this partial charge is going to like allow it to hang out with water, which is also polar, also has these like separation of partial charges. And so the threonine can now like hang out with water more than the ones that we've looked at in the past, which have only been like hydrocarbon chains. Today's post, we're going to use threonine as a way to talk about polarity, post-translational modifications, especially phosphorylation, and the importance of grad students, I guess. So we're at day 6 and we're talking threonine. So threonine, it might look kind of like the ones that we've been talking about, like it's like valine, except this alkyl, that is CH3 group of valine is going to be swapped out for an OH group. And so even though it's kind of like, it looks like it's kind of branched, we don't call it a branched chain amino acid, because it's not like a branched chain hydrocarbon. So in a hydrocarbon, you just have carbons and hydrogens. And so like these guys were all like, they all had just these hydrocarbon based chains. And threonine, it's going to have this OH group. And so this OH group is a hydroxyl group. And this is going to give it important properties. It's going to give it polarity. And it's also going to make it phosphorylatable. So basically when you have just like a pure hydrocarbon, so just carbons and hydrogens, well, that's pretty blah. But if you swap out the hydrogens for things like an oxygen or a sulfur or a nitrogen, that's where you get cool functionality, because these are going to make the molecules more reactive. So to understand why, just a quick reminder, so more on this in past posts, but all of these atoms that we're talking about being connected up, so all of these different carbons, hydrogens, and oxygens. So when I was showing you that model, like each of these balls represents an atom. So the black ones are carbons, the red ones are oxygens, the blue ones are nitrogens, and the white ones are hydrogens. So these are all connected up by sharing pairs of electrons. So basically each of these atoms is made up of smaller parts, protons, neutrons, and electrons. And so the protons are positively charged and they're held in the center. And then around them is this cloud of negatively charged electrons. And so these electrons are what they're moving around in this big cloud, and two atoms can kind of like merge part of their clouds to form a covalent bond by sharing a pair of electrons. So when they're sharing these electrons, because the electrons are kind of like in this cloud, you can think about sometimes if the two atoms that are joining together, if they both have like similar pulling power on those electrons, on their electrons. So the pulling power is coming from like the nucleus, the protons, the positive charge is kind of like raining in these negatively charged electrons. So atoms that are really good at raining in electrons, we call these electronegative. And so a great example of this is oxygen. So oxygen is really electronegative, so it can pull tightly on the electrons. So if it's sharing with something that is less electronegative, something that can't pull on their electrons as well, something like a carbon or a hydrogen, then they're going to hog the electrons that they're supposed to be sharing. And this is going to make the oxygen partly negative because it's getting more of this negatively charged electrons. And that's going to leave the other thing partly positive because it's like getting electrons pulled away from it, and the electrons are the negatively charged parts. So in this way, you can get molecules can have polarity, where you have this separation of charge. And so this, in the case of like water, and in the case of this OH group, this is like the molecule is neutral overall because it has an equal number of protons and electrons. But because you're having this uneven sharing, you can have like partially charged parts, and then these opposite charges attract, like charges repel. And so this water can form like these big networks, and they can also form like attraction, where they're being the like partial charged parts are being attracted to one another. And you can also have partially charged parts or fully charged parts from other molecules that water will let into its network. And so when you have like this separation of charge, we call something polar, and this allows water to be cooler with hanging out with water, or I guess water to be water to let it hang out with them because it has something attractive to offer. And so this is what we see when we have an OH group. But when you have a hydrocarbon, carbon and hydrogen share really fairly. So you don't see this polarity, you don't have polarity if you just have hydrogens and carbons. So if you have a hydrocarbon, this is kind of really blah in terms of like, both in terms of what the water would want to hang out with, which is why these are hydrophobic, so they're excluded by the water. And they're also blah in terms of there's nothing really to be attacked. Like there's no reason for another molecule to want to attack it, because it's kind of like boring. Like there's no vulnerable parts type of thing. So it can only undergo like combustion and stuff. But if you have a group like a hydroxyl group, this is going to give us the opportunity for nucleophilic attack. And this is going to give the opportunity to get phosphorylation as a post-translational modification. So these are a couple different concepts. So let's talk just first about post-translational modification and then how the nucleophilic part is going to come in. So a nucleophile is basically something that has more electrons than it wants, and so it'll attack something that an electrophile, which wants more electrons, and this is how you can actually form covalent bonds. So covalent bonds, remember, the strong bonds, like the bonds that are actually hooking together things. When we talk about like the polarity and stuff, that was all just involving like when we're talking about like attractions between molecules, like the water networks, those are all non-covalent bonds. So those are just attractions between like partial charge attractions. But with the covalent bond, you're actually like merging your electron clouds. You're actually forming these strong bonds. And so the oxygen, it basically has a lone pair of electrons. So it's going to allow it to act as go on like the attack, and this is going to be how we're going to get our phosphorylation, but we'll get more into this in a second. Let me just talk quickly about the idea of post-translational modification. So we call it post-translational because when you're making a protein, when the amino acids are getting pieced together based on the instructions of from the mRNA, this is called translation. And so if something happens to the amino acid after it's been added to the chain, then we call it a post-translational modification because it's happening after translation. So typically this is happening like after the protein's like fully folded, but you can also have it like before the protein's fully folded, and that sort of thing. So there are different types of post-translational modifications. So we'll see later when we talk about leucine that they can get like methylated and acetylated. The cysteines can form these like disulfide bridges. Prolines can get hydroxylated, but we're going to talk about phosphorylation today. Phosphorylation is where we have the addition of a phosphoryl group, and so we're going to add a phosphate group to our protein. We're going to add a phosphoryl group, and then there'll be a phosphate group. So basically, this phosphate group is where you have a phosphorus surrounded by 4 oxygens. And they can get transferred with the help of these enzymes called kinases. So enzymes are like reaction speeder uppers. And so a kinase can take this gamma ATP, so like this gamma phosphate, so this last phosphate group from the ATP. This is the same ATP that's like the RNA letter ATP and the same ATP that's used for energy. And it can add this phosphate group onto a protein, and we'll see how in a minute. But this can only happen at specific amino acids. And so the reason why it's like a phosphoryl group that we're technically transferring is because when we're transferring it, the oxygen is going to, one of the oxygens is going to come from the substrate. So from like the protein that's getting phosphorylated. And you can also have phosphorylation of like non-protein things like small molecules and stuff, but we're going to be talking about protein phosphorylation here. So we're going to add this phosphoryl group. So this like phosphate and 3 oxygens. But typically we just say like add a phosphate group. And yeah, but just if you want to be technical about it. So phosphorylation can only occur at specific amino acids because we need to have that like oxygen that's going to like attack the phosphate. And so it can happen at tyrosine, give you phosphotyrosine. It can happen at serine to give you phosphoserine, and that's what we're talking about today. It can happen on threonine to give you phosphothreonine. Because serine and threonine are both like kind of small and similar-ish size-ish, I guess, they can often, you'll have kinases that are like serine threonine kinases, so that can phosphorylate both. But typically like tyrosine, because it's like so big and bulky and stuff, it has like different kinases. So you'll talk about like tyrosine kinases and then like serine threonine kinases. You can also get phosphorylation of some other letters that are less common, like in bacteria and stuff. You can get like histidine phosphorylation, but in some mammals, but it's less studied because it's harder to study. And yeah. Yeah, so in the case of the histidine, you don't have the oxygen. Instead, you're going to have a nitrogen attacking. So you're really only going to get that phosphoryl group. And so you're going to have a phosphorus with like 3 oxygens and a nitrogen around it. But nitrogen's also like electronegative and stuff. And it has the lone pair, so it can act like oxygen in terms of being a nucleophile and attack. Anyway, so how does this actually work? And so the basics of it is going to be that we're going to take our protein and we're going to take ATP, and the kinase is going to help the protein attack the ATP to get the phosphate. And so the kinase, what it's really doing, so it's like the protein that's going to be doing the attacking. The kinase is just kind of like bringing to get them together, stabilizing things and making them interact. But it's this oxygen in that OH group that is going to be the thing that's actually going to do the attacking. And so this is why threonine's OH group is so important for this. And so basically, when you have this oxygen, it can act, it can get deprotonated, and this is going to make it a strong nucleophile. So a nucleophile is something that has like more electrons than it can comfortably handle. And so if it has more so we talked about oxygen really wanting electrons. Well, sometimes it kind of like gets more electrons than it gets, like the amount of the number of electrons that it wants to like fulfill its like octet. But so it has like a full outer shell. If you know what I'm talking about, cool. If not, look at past videos, or you don't really need to know that right now. But basically it has like the number of electrons it wants, but it has charge, and atoms don't really like having charge. So it wants to be able to share this, share electrons with another atom. So if you're sharing electrons from like a lone pair, you still get to like own those electrons kind of. So it's still in terms of fulfilling your cloud. So if this oxygen uses this pair of electrons to form a bond, it'll still count as having that pair of electrons when it comes into determining whether it's like cloud is filled, basically. So it's going to basically, it just wants to find something to share these electrons with. And the place where it can find them is an electrophile, which is something that wants electrons. So the reason why we call this a nucleophile is because it's seeking a nucleus. So a nucleus like is where the positively charged protons are, right? And so that's where it's going to get its help, which it can sharing that electron burden. And so nucleophiles are sometimes, but not always, negatively charged. And electrophiles are sometimes, but not always, positively charged. And these electrophiles are something that they have too few electrons or electron density. They want more electrons. And so you might be looking at ATP and thinking, well, this is negatively charged. How the heck is this supposed to act as an electrophile? Well, the oxygens are negatively charged, but the phosphorus, it's actually because the oxygen pulls the electrons away from it, the phosphorus itself is actually partly positive, and it's going to be electrophilic because it's having all of these electrons pulled. It has all these oxygens like pulling electrons away from it. So it's going to be electrophilic. And then we have the oxygen from our hydroxyl group. We're going to basically have the kinase is going to help deprotonate this hydroxyl group. And then this is going to make it a really good nucleophile. And so it's going to attack the phosphorus on the last phosphate group of ATP. And so basically a kinase is going to help make this happen. So when you have this hydroxyl group, we talked about before about how like the carboxylic acid group is typically like deprotonated. And this is because when you have this deprotonated, even though you have a negatively charged, which like atoms don't really like, this is actually resonance stabilized. So here the double bond is drawn to the oxygen, but you really have resonance stabilization, kind of like you have here between the nitrogen. So this is resonance stabilized. So basically the sharing of electrons between all three of these, and this makes them really happy. And so the carboxylic acid group is really acidic, and so it'll get deprotonated. But when you have just like a hydroxyl group, so just an OH group, this is typically going to be protonated. This is not a very good acid. So you're going to need a base to pull that acid to like deprotonate it. And so this base can actually come from the kinase. So in the active side of the kinase, there's an amino acid residue. So basically when the amino acids link together, because you're taking away the amino and the acid ends, when you join them together, you're joining through those ends. So they're no longer individual amino acids. We now call them residues. But we typically use the terms interchangeably. But so you have this, this residue sticking out into the active site. And this is with a carboxyl group. So you have this residue sticking out into the active site with an O minus. And then this is going to attack and make this an O minus. So then this becomes an OH and this becomes an O minus. And then this is going to be a really good nucleophile. And then your kinase is also going to be holding on to ATP. And in the kinase, there's going to typically be like magnesium ions that are going to help stabilize the ATP, because in the active site, so ATP has these like all these negatively charged things. And so the magnesium is positively charged, and it's going to kind of help stabilize things as this is happening, and shield the charges from one another. So once you deprotonate this, so this would be like a threonine or a serine or a tyrosine. So now this is a nucleophile, and so this is going to attack this electrophile, this phosphate. So you're going to make a substrate gamma phosphate bond and break this gamma phosphate beta phosphate bond. So when we talk about the 3 phosphate groups in ATP, it's like alpha, beta, gamma. So the farthest one is gamma. And so you're going to make this bond and break this bond. So this is just showing like the reaction mechanism. You got some electron pushing and stuff. So when you're attacking it, because then you're like, I can't form that many bonds. And so then this kicks off an ABP as you're leaving group, and you have a phosphorylated protein. So that is how the protein can get phosphorylated, and then this can influence how the protein acts and who it can interact with. And so I hope that helped you appreciate the biochemistry of threonine. And now let's talk about its discovery. Okay, so for this, I'm going to take you to my blog post because I need to look at my notes so I don't get anything wrong. And you can also find like this whole story there. So threonine was the last of these 20 amino acids to be discovered. And people didn't really know if there were more amino acids, like if was there a 20th, was there still a missing amino acid? So people were trying to basically figure out how many amino acids were, which of them were essential. So meaning that you need to get them from your food, your body can't make them from them, your body can't make them themselves. And so this William Rose and his grad students figured out that threonine is essential amino acid. And so how he's going to do this is by basically using his lab students as lab rats, but with their consent, like feeding them diets without threonine, and then they got all grumpy and had and stuff, and then he fed them threonine and they perked back up, and so he's like, it's essential. But anyway, that's the long story short. But the long story long, he didn't start with his grad students. He started with rats, and he wasn't the first person to do this. So in the 1910s, Osborne and Mendel fed rats with restricted protein sources. And so if they fed the rat's source food that was deficient in leucine, so the amino acid leucine, the rats would stop growing until you added back the leucine. Similarly with tryptophan. And so they're like, aha, these amino acids are essential. You need to get them from your food. And then in the 1930s, this American biochemist, who's the protagonist of our story, William C. Rose, he decided, okay, let's do things in a more controlled fashion, because these guys were just feeding things that they knew were low in this amino acid. But I want to know the exact components that you need. So I want to give these rats a controlled diet where I know exactly which amino acids I'm giving them. So he synthesized the amino acids. So he made them from scratch, or he purified them himself. And then he mixed and matched various amounts of them. So he knew exactly what was in there. So it's just controlled amounts of each. And so he modeled he didn't just wing it. He modeled what he gave the rats based on the amino acid content of the protein casein, which is a major protein in milk. So this was good enough if you added, the milk was good enough if you added histidine, but the synthetic mix that he made, which is supposed to mimic the casein, wasn't. And so he knew he was missing something. And so he suspected he was missing another amino acid. And so he set out to find what he called the new essential. So yeah, so it sounds like a beauty care product, but it was not easy breezy beautiful. Well, the end results were beautiful, but it was definitely not easy breezy. And so he started with 12 kilograms of the protein fibrin, and that he used acid to acid hydrolysis. So he basically added acid to split apart all of the different amino acids that were in the protein. And then he used tons and tons of different techniques to try to separate and purify out the amino acids. So he did a lot of selective extraction. So basically different amino acids or different molecules will dissolve in different solvents. So different liquids and stuff. And so you can use different solvents and see what dissolves in it, and to remove different components, and the things that don't dissolve in it will stay solid. And then you can separate fractions, and you can fractionate, and based on different solids. Basically, there's a lot of different techniques that you can use to try to tease apart these amino acids based on whether they're wanting to hang out, which liquids they like to hang out with, which liquids they dissolve in. And so he did this to remove the, so he used I know that there are these techniques I can use to isolate these specific amino acids and stuff, so I could get rid of these. So he basically got rid of all this known stuff, and then tried to figure out what was left in the stuff that he couldn't identify, basically. And so basically, if he needed to find what in that mixture there was that of unknown stuff there was, that if you added it to the known stuff, you could get the rats to thrive. So one of my favorite, in their paper, they actually say no procedure has been found which is not exceedingly laborious. So yeah, they told it like it was. This was 1935 when they were describing their discovery. So they had to one of their steps involved extracting 17 times with 40-liter portions of butyl alcohol. Wow. Okay, so basically they did a lot of work, and they ended up with about 4.5 grams. So about 0.6 grams per kilogram of that fibrin protein that they started with. And this was in a pretty crystal form. And crystal is really good when you're trying to purify something, because something has to be when purification is crystallization is actually a purification technique, because all the it means that all of the molecules have basically arranged themselves in this orderly way to give you, and they have to be pure in order to do that, or else there's other stuff in there, then it won't be able to form this nice crystal. So they have this pure leucine. Yay. Okay. And so the data from their paper, the data demonstrate conclusively that the crystalline compound is the new essential we have been endeavoring to isolate for several years. Furthermore, the experiments reported in chart 1 represent the first successful efforts to induce growth in animals upon diets carrying synthetic mixtures of highly purified amino acids in place of proteins. Yay! So why so exciting? So they weren't just, it wasn't just that they discovered what there was this molecule here. They also showed that if you added just this molecule to the other essential molecules that you needed, then these rats were living normally. So they had found like that missing thing, that they now had a defined diet that they could give the rats in order to like get the rats to live. And so now they knew how they could, like, what was needed, what was essential. But this was hardly the end, because they isolated this thing and they're like, yeah, we got crystals, we have this pure thing, but they still didn't really know what it was chemically. So they had to do a lot of chemical experiments to figure out like what it would react with, trying to make other things with it and characterizing those things, and then like inferring back like, okay, so if we know we can make this from it and this has this property, so the original thing must have this property. But so eventually with that, and they were able to determine it would be an alpha amino beta hydroxybutyric acid. So butyric, so it has like that 4 carbons, and then like it's on the alpha amino, and then the beta, so the alpha carbon on the beta carbon, so the next carbon, there's the hydroxyl group, and you have the carboxyl group, the amino group. So they figured out what the side chain was, but they didn't know like which way it was sticking out like stereochemically. So in terms of like, we've talked about like chiral centers. So basically threonine, like isoleucine, but unlike all the other amino acids, it has a second chiral center. So it's chiral centers where you have a carbon attached to 4 things, and they can attach in like sticking out in different ways. And so you can have different stereoisomers, which have like all the same like atoms linked in the same, like this OH group is linked to this part, but it can be sticking like forward or back, basically. And they didn't know if it was sticking forward or back. But so this one has, all amino acids have that at this, their central carbon, and all of them are in this like L isoform. But threonine also has a chiral center in its side chain, and so they didn't know which one this one was. And so the answer to this didn't come until later, when H.E. Carter synthesized all 4 options and showed that the natural one corresponded to 2S, 3R, 2-amino, 3-hydroxybutyric acid. Butyric acid. Yeah. Okay. But anyways, so now they figured out it was essential. And they had a full set of amino acids that they could make a totally artificial diet, protein-wise. And then they could exclude these amino acids one at a time and see what happens. And they could see just how much of each one was required for rat healthfulness. So they could do all this experiments with rats, but well, is the same true for humans? And so rats weren't really human-like enough. And so he next turned to dogs, and he got similar results. But dogs still aren't human. And so if he really wanted to know whether this was essential for human, and whether the other ones he thought were essential really were essential for humans, he needed to turn to humans. And so that's where these grad student volunteers came in the 1940s. And so when he didn't give them threonine, they only experienced like minor symptoms like irritability, fatigue, loss of appetite. And this only happened when certain amino acids were withheld. These were isoleucine, leucine, tryptophan, lysine, methionine, phenylalanine, threonine, and valine. So he said, okay, these amino acids are essential, whereas the rest can be made from other things in our bodies. And then when he fed the students normal food again, they perked back up. So if you want to learn more about this, you can check out this like JBC article. And it also has links to like the original papers and stuff. So it's a pretty fun read. Yeah, so in terms of codons, this is all that information. As for what in terms, it's like ACU, ACC, ACA, and ACG. So it starts with AC. If your codon starts with an AC, you get a T. So yeah, so hope that helps you appreciate all of this.